everybody had a good break. Hope everybody got their exam. Hope everybody's happy with their exam. No. So the average on the exam was 65, and that was a significant improvement over before. I don't think I've posted the curve yet, have I? No, okay. So I will get that posted shortly, and you can see where you stand with respect to everything, getting ready for the final. So the final is in this room, I believe, on Thursday of finals week at 6 p.m. Yeah, I'm no happier about it than you are because I have to have grades in by Monday, and that's a lot of grading between Thursday and Monday. So hopefully we get it all together. Well, we are moving rapidly through the material, and we are finally at the topic of glycogen metabolism. And so you're going to like glycogen metabolism, and you're going to dislike glycogen metabolism. Okay? It's a great intro to a topic. What you will like about glycogen metabolism is you will start to see, excuse me, I caught a cold over the weekend. You will start to see integration of pathways. You'll see how glycogen metabolism is tied to glycolysis and gluconeogenesis. So you start to get a better picture of the complexities going on in the cells of your body. And you'll see that as a metabolic pathway goes, glycogen metabolism is the simplest pathway you will ever study. It's the simplest pathway you'll ever study. You'll see about 3 steps, and that's it. Okay? There's really not much to it. Now, what you won't like about it is the regulation is complex, but very interesting. Okay? Very interesting. So, hopefully, with that warning, everybody is now suitably prepared for glycogen metabolism. Okay. Well, let's talk a little bit about glycogen itself before we get... Actually, let's do a limerick before we get started. Out at State Farm, the agent extends a large payment to tie up loose ends. So this brings to a close what the owner now knows, all the ways that a Mercedes bends. Okay. All right. Now, we are ready to understand, hopefully, something about glycogen. So this schematically shows the structure of a portion of a glycogen molecule. Glycogen molecules are very large. They are, if you recall from the earlier lecture, a polymer of glucose. Glycogen contains only glucose, and it contains glucose with 2 kinds of linkages. It has, most of the linkages are in alpha 1,4 links, and that consists of all of the bonds that are going across this guy, as you see on the bottom. There are also the bonds that are across this guy going up on the top. The only place where we have other bonds or where we have branches between the two. So the alpha 1,6 is the connection between individual chains of alpha 1,4s. Okay? That's the only place where we see something besides a 1,4 bond. That's the linkages between the individual chains of 1,4s. They're linked by a 1,6. And you can see here, for example, this glucose has a 1,6 linkage, and it also has a 1,4 linkage on the other end. Okay? Now, I mentioned earlier that glycogen is interesting in its structure when we compare it to amylose or if we compare it to amylopectin. And I'll remind you what those are. So amylose is a polymer of glucose that's found in plants. Plants use it to store glucose, like your cells use glycogen to store glucose. And amylose is a polymer of glucose units, all alpha 1,4. Everything is alpha 1,4, meaning no branches. It has 2 ends, a beginning end and an ending end. Okay? And that's all that there is to an amylose chain. It can be very long, but it only has 2 ends. Amylopectin is similar to glycogen in that it has alpha 1,4 bonds, and it has alpha 1,6 branches between individual chains of alpha 1,4 bonds. The difference between, excuse me, amylopectin, and amylopectin is also a plant polysaccharide. Okay? The difference between amylopectin and glycogen is that amylopectin has branches about every 50 or so units, okay, each unit being a glucose residue, whereas glycogen has branches about every 10. that means that glycogen has many more branches than glycogen does. And you might say, well, it's got 5 times more branches. It actually has more than that because each chain itself can have branches. There's a lot of branching that's going on in glycogen. Well, the more branches that you have, the more ends that you have. You have one starting glucose, but you have thousands of ending ones because the end of every branch is a glucose. Sorry. As I mentioned earlier, that's very important for animals because animals need to break down glucose quickly because they need energy for muscles to contract and run and jump and do all the things that animals do. Okay? And so it means that because the enzymes of glycogen start at ends and work inwards, okay, that there are many, many more ends to work on. That means much more glucose can be released at once from glycogen than can be released from either amylopectin or from amylose. Okay? Now that turns out to be important, again, because animals have these quick energy needs. Plants don't have quick energy needs like that, so they don't need so many ends to release a lot of glucose quickly. Their needs are fairly constant. Okay. I kind of like that. That is a schematic way of representing glycogen where you have a starting point and then all the branches that come out and off. Okay? That's how you might envision glycogen actually looking. Okay. Well, this is actually how you see glycogen inside the cell. There are glycogen granules that are these little black things that are there. They've been stained for you to be able to see those. And they're abundant, as you can see. Okay? And if we look at what happens to glycogen, we're first going to look at its breakdown. The breakdown of glycogen occurs through an enzyme called glycogen phosphorylase that we'll talk a lot about. And glycogen phosphorylase cleaves alpha 1,4 bonds. It cleaves alpha 1,4 bonds. And when it cleaves alpha 1,4 bonds, it produces glucose 1-phosphate. Okay? Well, glucose 1-phosphate, of course, is not an intermediate of glycolysis, but as I mentioned in the lecture a couple of lectures ago, glucose 1-phosphate is readily converted into glucose 6-phosphate by the enzyme known as phosphoglucomutase. Can you tell me the name of the intermediate of this enzyme? There's a point of extra credit. Yes? Glucose 1,6-bisphosphate. Yes. How did you know that? Right. So mutases work by having a diphosphate intermediate. It puts on a phosphate, then it takes off the other phosphate. So the intermediate in the action of this enzyme is glucose 1,6-bisphosphate. And yes, cells do release glucose 1,6-phosphate like they released 2,3-BPG, which was the other mutase enzyme that we talked about. Glucose 1,6-bisphosphate, however, doesn't signal anything in the cell. Okay? But glucose 1,6-bisphosphate is released as a result of action of this enzyme. Well, the product of that reaction is glucose 6-phosphate. And that turns out to be very useful because that glucose 6-phosphate is an intermediate in several pathways. You've only seen one so far. The pathway we've talked about so far is that of glycolysis. Glucose 6-phosphate can go to pyruvate and produce energy. If we're in the liver and the liver needs to put glucose out into the bloodstream, glucose 6-phosphate is readily converted into glucose by simply clipping the phosphate off. You saw that in gluconeogenesis. And last, one pathway we haven't talked about is the pentose phosphate pathway. This is a pathway where sugars are converted into other sugars. And specifically, what happens in the pentose phosphate pathway is a common end product, a common end product of the pentose phosphate pathway is the production of ribose 5-phosphate. Ribose is, of course, needed for nucleotides. Okay, so that shows the sort of utility of glucose 6-phosphate. Now, the enzyme glycogen phosphorylase, as I said, I'm going to spend a fair amount of time talking about because glycogen phosphorylase is interesting not just in its reaction, but also in its regulation. Okay, the regulation of this enzyme is complex, and it's very cool and interesting. Well, before we talk about its regulation, let's talk about its catalytic action. So the catalytic action of the enzyme is also interesting because look at what it does. Here's a glycogen molecule, and here's an alpha 1,4 bond, and this guy is at the end because you can see that the 4-hydroxyl is free. It's not linked to something else. So we're at an end. There's a 1,4 bond right there. Okay? And the enzyme uses a phosphate to cleave this bond and produce glucose 1-phosphate. Now, that's a reaction like you haven't seen before. It's a reaction we call a phosphorylasis. A phosphorylasis, okay? A hydrolysis uses water to break things. A phosphorylasis uses phosphate to break things, okay? Well, this turns out to be important, that it's using phosphate. What significance would that have from an energy perspective? Why doesn't it just use hydrolysis and cleave to give a glucose? Any thoughts? That's right. The product of this reaction has more energy, and we didn't use ATP to put the phosphate on. Remember, if we started with glucose and we want to make glucose 6-phosphate, which we can make from glucose 1-phosphate readily, if we wanted to make glucose 6-phosphate, we had to use hexokinase and ATP to put the phosphate onto glucose to do that. That tells us that putting that phosphate onto glucose required energy. Everybody with me here? Okay. So the cell has saved energy by using phosphate instead of ATP to break this bond. Now, the question that should immediately arise in your minds is, well, if it costs energy to put a phosphate onto this molecule, how in the world does it put the phosphate on there without using ATP in the first place? Thoughts about that? Well, you guys had too much turkey, didn't you? Too much turkey. How about the fact that the alpha 1,4 bond has some energy in it? And when that bond is broken, the energy of breaking that bond is used to attach the phosphate. The energy of breaking that bond is used to attach that phosphate. So the cell is very efficient. The cell uses energy sources wherever it can. And the energy of breaking that bond allows that phosphate to be put onto it. That means, therefore, that when we go to make that bond, this bond right here, when we go to make this bond in the cell, we're going to have some energy input to making that bond. And we'll see about that as we get to the enzyme known as glycogen synthase. Okay? So this is, and so the product of that reaction, of course, glucose 1-phosphate is one of those, and then glycogen that has lost 1 glucose. Because we have so many ends, we can produce many of these very quickly, and the glycogen can shrink pretty quickly. One of the reasons that cells have such elaborate controls on their glycogen metabolism is that it allows the cell to respond very quickly to energy needs, and it allows the cell to not break down things too fast that it doesn't want to break down. So it's important that the cell have both quick and slower controls for breaking down glycogen, and we'll see how that happens. Okay. Well, it turns out that glycogen phosphorylase is not the perfect enzyme. Okay, it's certainly not a perfect enzyme in our definition of a perfect enzyme, but it's also not a perfect enzyme in that it doesn't by itself completely break down glycogen. Glycogen phosphorylase only works on alpha 1,4 linkages, it won't touch alpha 1,6 linkages. And more complicated than that, it won't touch alpha 1,6 linkages within about 4 residues of a 1,6. So if we had the structure that you see up on the top, and we treated it with glycogen phosphorylase, what it would do is it would clip off all of these red guys, but it would not touch the yellow or the blue because they're within 4 or 5 residues of a branch point. Okay? And no, I don't expect you to memorize this structure, right? But you should know that this enzyme, glycogen phosphorylase, will only work within about 4 or 5 residues of a branch. If we wanted to break down the glucoses in there, another enzyme is required. Okay? So cells have another enzyme that will catalyze the reaction that you see on the screen. It actually occurs in 2 parts. The first part is the transfer of 3 of the glucoses to the end of the other chain. So you see the 3 blue guys here moving down to the end of the chain. So we're breaking an alpha 1,4 bond, and we're making an alpha 1,4 bond. Right? No energy difference. We don't need energy to make that happen. Okay? Then something surprising happens. The same enzyme, and by the way, they label these as different activities, but these are the same. They're both activities in the same enzyme. Okay? The enzyme name I give you is much simpler than the one that most people call this enzyme. The name that what most people call this enzyme is about this long, and I can never remember it myself. So I call it debranching enzyme. And you can call it debranching enzyme. Okay? Debranching enzyme catalyzes first the transfer of the blue down to the lower chain, and then it uses water to break this bond and release a free glucose. Now, based on what I told you earlier, you might ask the question, why doesn't it use phosphate to release glucose 1-phosphate? Why doesn't it do what glycogen phosphorylase does? Any thoughts? Nobody wants to think today. Back there. I can't hear you. There's not enough energy in an alpha 1,6 bond to make that happen. That means there's less energy in an alpha 1,6 than there is in an alpha 1,4. Understand? That's where the point of extra credit. Give me a holler later, okay? All right, so the upshot of this is now, after this debranching enzyme has worked, what happens is we have free glucose. That's the only place that free glucose is released in glycogen metabolism. And we have a substrate that glycogen phosphorylase can say, oh, no branches. I could come clip, 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 clip until it gets within 4 of a branch again. The combination of these 2 enzymes, along with phosphoglucomutase, which converts glucose 1-phosphate into glucose 6-phosphate, is the entirety of glycogen breakdown. That's all there is to it. 3 enzymes. Okay? I told you it would be a simple pathway. There's the glucose 1,6 intermediate that you see there. We talked about that before. Not a biggie. And we're set. Okay. There's the structure of glycogen phosphorylase, if you wanted to memorize that in any way. Glycogen phosphorylase is an interesting enzyme, as I said before, because glycogen phosphorylase exists in both a T state and an R state, and it is covalently modified. Okay? So that's something we're going to spend some time talking about. So it exists in a T state, it exists in an R state, and it's covalently modified. And so the upshot of that is that it turns out that glycogen phosphorylase can exist in 4 different forms. Okay? The covalent modification that occurs to glycogen phosphorylase is a phosphorylation. Our friend protein kinase A that we activated in the epinephrine pathway, protein kinase A activates an enzyme I'll describe in a little bit that phosphorylates glycogen phosphorylase. That puts a phosphate on. There's a phosphoprotein phosphatase that takes the phosphate off, and I'll show you both of those later. The upshot of that is that there's 4 forms of glycogen phosphorylase. A form that's in the R state with the phosphate on, a form that's in the R state with the phosphate off, a form that's in the T state with the phosphate on, and a form that's in the T state with the phosphate off. Okay? The forms that have the phosphate are called glycogen phosphorylase A. The forms that lack the phosphate are called glycogen phosphorylase B. And by the way, many books, including your book, refer to glycogen phosphorylase A as simply phosphorylase A. I prefer that you use the term glycogen phosphorylase A, or glycogen phosphorylase B for that matter. Okay. All right. Now, here are the 4 states of glycogen phosphorylase. So as I said, we have a form in the B form, lacking phosphate. It can exist in an R state. It can exist in a T state. We have an A form, that is the form that has the phosphate, and you see this is a dimer, so each of the monomer units gets a phosphate. The phosphorylase A can exist also in the R state on the top and the T state in the bottom. As we go from left to right, on either the top or the bottom, we're putting a phosphate on. Putting the phosphate on requires action of an enzyme known as phosphorylase kinase. That's an important enzyme, as we shall see. Phosphorylase kinase. Hi, are you here from the CMC? I've actually got it straightened out, so thank you for checking by, okay? We go from the unphosphorylated to the phosphorylated. Requires the action of phosphorylase kinase. If we go from right to left, that means removal of phosphates, and that requires the action of an enzyme known as phosphoprotein phosphatase. Okay? As we go up and down, how do we go from R state to T state? I'll ask you that question. How have we seen that we go from R state to T state or T state to R state for an enzyme? What was required? Not covalent modification, no. Only allosteric binding. Covalent modification does not change R and T. Okay? Allosteric binding is what changes R and T. So that means that as we're going left or right, we're talking about covalent modification. If we're going up or down, we're talking about allosteric modification or allosteric binding. Okay? Okay. All right. Now let's flesh this out a little bit. Here is, and by the way, your book will talk about this occurring in muscle versus liver, etc., and I find that if I start making it tissue-specific, students get confused. So we're going to treat it as if it's all the same, although it's really not all the same. The important thing here is understanding the overall picture of the regulation. So you can just blot out your mind that it says muscle, and the next figure where it says liver. Okay? Here is what's going on. Okay? In muscle, we have a T state to an R state. Okay? We have phosphorylase B. So the fact that it says phosphorylase B means we're only talking about the unphosphorylated form here. So we're following the allosteric changes of the enzyme, okay, in the unphosphorylated form. All right. As we go from left to right, we're going from T state to R state. R state is the more active form. T state is the less active form. Glycogen phosphorylase B is in the most active form when AMP is present, because that moves it from left to right. Right? What's AMP an indicator of? Low energy, right? Low energy means the cell is saying, I need energy, It's activating glycogen phosphorylase instantaneously. Allosteric modifications occur instantaneously. It doesn't take any time at all for them to happen. AMP binds to the enzyme moves to the R state, and it starts breaking down glycogen and producing glucose 1-phosphate, which can then either be made into glucose and be exported or made into glucose 6-phosphate for oxidation. With me? What converts the enzyme from the R state back into the T state? Okay. Well, there's two things that will do it. Okay. One is ATP. What's ATP an indicator of? High energy. Okay. If we have plenty of energy, do we want to be breaking down our glycogen? No. Do we want to stop that breakdown instantaneously? Yes. Okay. Glucose 6-phosphate is also there. What's glucose 6-phosphate an indicator of? That's a harder question, isn't it? Glucose 6-phosphate is actually an indicator of the fact that glycolysis is plugged up, or that the cell is making glucose. Either of those is true. Okay? If glycolysis is plugged up, that tells this enzyme the cell doesn't need energy because it's not breaking down the glucose that it has. And so again, it instantaneously turns this enzyme off. Okay? Now, it turns out that most of the time inside of our cells, there's very, very little AMP. What does that mean about the glycogen phosphorylase B? It means it's mostly in the T state. And because it's mostly in the T state, people talk about glycogen phosphorylase B as if it's the less active form of glycogen phosphorylase. That may not be totally accurate, but you can see why that would happen. If you have a form of the enzyme that is inactive most of the time, you would probably describe it as a less active form. However, it is possible for glycogen phosphorylase B to become active, and that can occur when AMP is present. So if you're heavily exerting yourself, you're very quickly burning up your energy, you can activate glycogen phosphorylase B, and there are times where that may happen. Let me give you an example. Okay? Imagine that I use my favorite scenario, the building catches on fire and we decide we have to get out of here and we have to run quickly and get away. Okay? Our adrenaline starts flowing. We've seen one thing that will give our cells ATP that was a buffer. And remember the molecule I'm talking about? Creatine phosphate. Creatine phosphate would put ATP available to the muscle cell instantaneously. Okay? Creatine phosphate is not an infinite resource. When the cell starts running out of creatine phosphate, or ATP from creatine phosphate, its ATP levels will start falling, and that may happen before the epinephrine makes it to the target cells. Wouldn't it be nice to have a backup that would release glucose before the epinephrine got there? This is it. If AMP levels were to rise before the epinephrine kicked in, instantaneously this guy is going to become active, and it's going to start, the cell is going to start producing glucose 6-phosphate, or glucose 1-phosphate to convert into glucose 6-phosphate that it can use for energy. So this instantaneous allosteric activation turns out to be very, very useful. Our body has a lot built into it, okay, to allow us to handle almost any need that we have. When epinephrine kicks in, of course, it's going to now favor some things that will convert the B form into the A form. We'll see how that happens. Okay. Questions on that? I don't want to jump ahead too far there. Questions? Garrett?
Yeah, what happens the chains of like three molecules? What happens to like that chain? What happens to that?
What happens to the what? Okay, hold on. I'll go back there. This one?
Yeah. What happens to like that?
What happens to this guy? It's broken down into individual units because the enzyme is clipping them off one at a time until it gets within 4 residues of a branch. So this right here is glucose 1-phosphate, right? So as long as it's more than 4 units away from a branch, it'll keep clipping the individual units off. Okay? Justin.
Can glycogen phosphorylase not act within 4 residues of an alpha-6 or alpha-1,6 linkage because of steric hindrance? Or is there another?
Yeah, so the question is the limitation on glycogen phosphorylase not working within 4 units due to steric hindrance? And that is indeed the limitation, that the structure is such that it can't handle that structure. Other questions? Okay. Yeah, up top.
So where in that diagram is debranching enzyme acting?
Debranching enzyme is acting in 2 places. So the question is, where in the diagram is debranching enzyme acting? It's acting in 2 places. One, it's taking the blue guy down here. That's what this thing refers to as the transferase. That's the transferase part of the debranching enzyme. And the second is it's clipping this glucose off right here. That's what this calls the alpha-1,6 glucosidase, and that's that part of the debranching enzyme. So both of these activities are part of the debranching enzyme. Okay. All right. So now we've seen the activation and inactivation of glycogen phosphorylase B allosterically. Let's do the same thing for glycogen phosphorylase A. Glycogen phosphorylase A is interesting. I keep saying interesting. That's my favorite word of the day, I think. It's interesting, guys. All right, as we look at glycogen phosphorylase A, we remember, first of all, that glycogen phosphorylase A has phosphates. So in both cases, we see phosphates on either side. We're not covalently modifying this guy. It's already been covalently modified. Okay? And if we look in going from left to right, we see nothing. There's no allosteric activator of this enzyme. What that means is this enzyme will spontaneously, all by itself, flip from T to R if glucose is not present. So long as glucose is not present, this enzyme will exist in the R state. That means that most of the time, remember glucose is a poison, that means that most of the time this enzyme exists in the R state. And that's why many people describe the glycogen phosphorylase A as being the more active form of the enzyme. Okay? Describe it as being the more active form of the enzyme. It's usually in the R state. What causes it to go into the T state? Well, only when glucose accumulates does the enzyme go into the T state. As glucose accumulates in the cell, the enzyme flips from the R state to the T state. All right, so when would this happen? When would glucose accumulate in the cell? Well, it depends on the cell, of course. It could be a muscle cell, it could be a liver cell, so we don't have to worry about whether it's muscle or liver up here. Glucose will accumulate in a muscle cell when the cell is not burning glucose. It's not going through glycolysis. So just like glucose 6-phosphate will accumulate, so too will glucose. And when glucose starts to accumulate in the cell, all right, we don't want to be breaking down our glycogen and producing even more glucose 1-phosphate, so the enzyme flips from the R to the T. If I'm a liver cell, what's a liver cell doing? Liver cell is modulating blood glucose levels, meaning it's trying to control them. Well, if the body has been exercising and the Cori cycle has been running, then the liver has been dumping glucose into the bloodstream, as we saw in the Cori cycle last time. When the bloodstream quits taking up glucose, what happens to the concentration of glucose in the bloodstream? It goes up, right? When it goes up, the liver senses that and it stops putting glucose out. When it stops putting glucose out, what happens to the glucose concentration inside of a liver cell? It goes up. And what happens to the glycogen phosphorylase? It gets turned off. Okay? So again, we have elaborate controls, and this control is also instantaneous because this is an allosteric interaction, instantaneously turning glycogen phosphorylase from the R state into the T state. Question? Okay. Question, if you're exercising and you stop and your blood glucose levels start to rise, will that cause release of insulin? I can't tell you definitively, but it wouldn't be like the glucose rush that you would get from drinking a Pepsi Cola. Okay? So you might see a little bit of a rise, but it wouldn't be anything like what you would get if you were drinking or eating glucose. Okay? Okay. So one of the reasons that doesn't happen is your body has, as you can see, controls that kick in almost instantaneously to stop something from happening to excess. And that's a very important consideration. Okay. So that relates to the regulation of glycogen phosphorylase. Oh boy. Now I said that the enzyme that puts the phosphate onto glycogen phosphorylase is known as phosphorylase kinase. It's also called glycogen phosphorylase kinase. I'll take either term. Okay? Phosphorylase kinase exists in two forms. It exists in an inactive form, and it exists in an active form. And it also exists in a partially active form. So I guess there's three forms. It's not quite as confusing as it might seem. Okay? Let's talk about one of the controls. So you can see that this enzyme is also controlled by covalent modification and by allosteric interaction. Okay? Here's the inactive form of phosphorylase kinase. Remember, this is the enzyme that puts phosphate onto glycogen phosphorylase B to make it glycogen phosphorylase A. Okay? The inactive form has no phosphate on it, and it's not bound to calcium, which is the allosteric activator. No phosphate on it, no calcium. How does it get a phosphate? Our friend protein kinase A puts the phosphate on. So if I have epinephrine that's being released in the cell, released by the body, and binds to a target cell, it binds to the beta-adrenergic receptor. It causes the beta-adrenergic receptor to activate, ultimately releasing cyclic AMP, activating protein kinase. Protein kinase, one of the things it does is it puts phosphates onto phosphorylase kinase. That converts the phosphorylase kinase from the inactive form to the partially active form. Okay? If calcium is present, the partially active form becomes fully active because the enzyme binds to calcium. The top pathway occurs relatively slowly because it takes time for the beta-adrenergic receptor to start activating everything to make protein kinase A be active. The top pathway is relatively slow. Okay? The bottom pathway, as you might guess, starts out very quickly. Inactive phosphorylase kinase becomes partly active in addition by the binding of calcium. What cells release calcium as a way of doing what they do? Muscle cells. When muscle cells contract, they release calcium. Calcium concentration rise, and a sign that calcium concentration is rising in a muscle cell means I'm contracting, I'm going to need energy. The muscle cell has an immediate source of activating phosphorylase kinase, just through releasing the calcium that it does for contraction. Okay? This occurs instantaneously. Even before the hormones make it, that is, the epinephrine makes it to the target cell, even before that happens, this phosphorylase kinase has started to be active. Okay? We've instantaneously started the activation so that we can break down glycogen for these muscle cells that are contracting. To be fully active, of course, this cell has to wait for the epinephrine pathway to kick in, but it's actually got a jump start on it by just the partial activation that you see down here. To be fully active, I'll remind you, it takes both phosphorylation and calcium to be present. When that happens, phosphorylase B is converted into phosphorylase A, and phosphorylase A, of course, is what we describe as the more active form. Everybody with me? Questions on that? Question over there, yes? Does binding of calcium affect the phosphorylation of this enzyme? You know, I don't know. That's a good question, actually. You might expect that this might be a slightly different shape than this is, and that you might see a difference in the... Not good. You might see a difference in the activation of that, and I don't know the answer to that. There might be a slight difference between the two. Okay, let's see. How am I doing? Okay, I think I'm at a point. I'll tell you, let me just show you one more figure, and then I'll review this figure next time. All right? Oh, wrong one. This figure now completes the pathway that we started earlier with the beta-adrenergic receptor. So I just want to briefly step you through it. The body has released epinephrine. Another molecule that I haven't talked about, but is equally important, is an enzyme, is a molecule called glucagon. Glucagon in the liver has the same effect as epinephrine. That is, binding of it to the receptor excuse me, will stimulate the exact same pathway that epinephrine stimulated. I like to think of glucagon as meaning glucose all gone. Okay? Glucagon is the liver's way, and it's specific to liver, it's the liver's way of responding to low blood glucose concentrations. It means, therefore, that it doesn't take fright or being scared to activate the beta-adrenergic receptor pathway. The liver can do it in low glucose concentrations. Glucagon is a peptide hormone. Insulin was a peptide hormone. Glucagon is a peptide hormone. All right, so in either case, the beta-adrenergic receptor fires. It causes the G protein to replace GDP with GTP. That activates adenylate cyclase. Adenylate cyclase makes cyclic AMP, which activates protein kinase A, which phosphorylates phosphorylase kinase, which phosphorylates phosphorylase B to phosphorylase A, and now we're breaking down glycogen. This is why I told you earlier that that pathway through the release of epinephrine results in increasing blood glucose. Okay? Questions on that? Shall we sing about it? This is a song to the tune of Raindrops Keep Falling on My Head. Brain farts just happen in my head. I think it might be due to something Kevin said. Biochemistry gets brain farts a-popping in my head, and they're popping. So I just wiped out the teardrops from my eyes and told my brain it had to do some mental exercise, burn some ATP, so brain farts can stop inside my head. They'll be stopping, 'cause there's one thing I've learned. When energy increases, it sure pleases my mental state. I'm doing great as tension eases. Da-da-dum. Now, brain farts don't happen in my head, so I'm sure the final will be easier. Instead, cyclic AMP stops brain farts from popping in my head. They're not popping thanks to caffeine Nothing's worrying me.